Hi everyone. Thank you for joining this week's Single Cell Spatial Exploration Club. I'm quite excited to show you some of the updates we've had in the SmartBulk application, especially for any researchers that are joining the call that are specifically concentrating on bulk RNA-seq pipelines and also being able to run other types of analysis on public data sets that previously might have not performed deconvolution, gene network analysis, and other sort of functionalities as well. So that will be the main focus today. We'll be sort of concentrating on a very specific case study, which relates to patients that have received sort of stem cell transplantation treatment or during sort of acute injury and sort of developed respiratory infection following that post stem cell transplantation. So this will sort of be the focus. We'll try to use some new features within SmartBulk to really deep sort of go into a deep dive into cell compositions within the data, as well as if we're able to really identify different gene networks that are driven by modules specifically within these different patients. So some of the patients that we'll sort of go through is sort of acute respiratory distress syndrome, patients that also have sort of acute respiratory distress syndrome, but post sort of this hematopoietic stem cell transplantation, and also looking at control sort of samples relating to sepsis, which is another sort of form of dysregulation in the immune response. So that will be the focus. So I'll go through our next slide here. So the title of the session today will be sort of how can we leverage smart bulk, specifically looking at bulk transcriptomic deconvolution and gene network analysis to really reveal if we're able to identify very distinct immune dysregulation post this hematopoietic stem cell transplantation, and particularly how this can lead to a different profile of acute lung injury. So that will be sort of again, the aim of the session. Before I go into sort of what the case study that we're focusing on this week, I just wanted to recap the session that we previously sort of conducted in the single cell sort of exploration club, which focused on the Tahu 100 million perturbation atlas. So if there is an interest within anyone who's joining the sort of exploration club today, please feel free to sort of contact us if you're interested in exploring this specific atlas to look at the sort of single cell perturbation and how this can be context dependent in gene function. So it will include 50 cancer cell lines, over 1,100 drug dose perturbations, and other analysis that you can really perform using bBrowser X when analyzing this specific data set. So just a recap of what we were able to really find in terms of the analysis that we performed on this atlas. So we were able to identify we were able to really answer if darfinibin, which is a different treatment, shows similar downregulation in other cancers. So not only sort of cancer specific. So we did see that in breast cancer, there is an absence of RAF and other mutations. But when it came to the specific treatment, it still showed some effectiveness within this sort of signaling pathway. So really the ability to be able to go deeper into the specific data sets and answer your own specific research questions and other functionalities. If you're interested again in this, we do have the recording of the previous episode in case you do want to sort of analyze this public atlas. Okay. So one thing that I do want to sort of cover before we dive deeper into the case study is really going into what acute respiratory distress syndrome is as sort of a disease model, how that relates to hematopoietic stem cell transplantation, and what are the current clinical challenges and molecular opportunities that we see within this disease space. So acute respiratory distress syndrome, as just sort of the condition itself, it's sort of known as a severe and often fatal complication, especially in patients with critical illness in the ICU. So in the intensive care unit. And normally the profile of these patients is marked by profound sort of hypoxemia and also cardiogenic pulmonary edema. There's a lot of vulnerable patient populations, especially within this context, and recipients of this sort of stem cell transplantation that face a particularly higher risk when it comes to mortality. So if we sort of focus on these individuals, not only sort of in the acute respiratory distress syndrome sort of case, so just the general triggers that we see within these sort of acute lung injury, and other sort of conditions, we can really dive deeper into what are the molecular mechanisms that drive these complications, especially when it comes to idiopathic forms of pneumonia, alveolar hemorrhage, or other sort of comorbidities that are present in these patients with ARDS. So there's a lot of recent evidence and recent research that really shows that patients with ARDS that also undergo sort of hematopoietic stem cell transplantation may have very distinct immunopathologic mechanisms that really differentiate these patients from what we see in normal sort of ARDS patients in the ICU. And there was recent studies, particularly working with bulk RNA-seq, that really uncovered a suppressed interferon response that altered this sort of immune gene expression within this specific group that undergoed this allogenic sort of transplantation treatment. However, there's not many studies that really focus on not only sort of differential gene expression analysis, but really going into the regulatory architecture within the data, looking at co-expression networks, cellular composition by using sort of deconvolution methods, and really seeing if there's any influence in terms of treatments that are used within these different patient groups. So that's really a bottleneck currently within this space. So sort of our main aim is really how can we use Smart Bulk as an application for your own sort of bulk RNA-seq analysis to really dive deeper into not only single gene differential gene expression, but really capture if there's any network level regulation across these different patient groups. Are there any treatment-driven transcriptional modulation that we can see when we perform an additional enrichment analysis from those sort of networks? And does that give us some indication on sort of this dysregulation, I guess, characteristic within this specific patient group that has a higher risk of mortality? So that's really sort of a background, so you get an idea what sort of analysis we'll be performing. And the agenda for today will mainly focus on three different aims. So the first aim will be really how can we identify co-expression modules driving this sort of acute respiratory distress syndrome pathogenesis post sort of this transplantation treatment. This will be using sort of the WGCNA method, which is the weighted sort of gene cell network analysis method. The second sort of aim will be how can we quantify and compare these different immune cell type compositions from bulk RNA-seq data? So can we use a public sort of data set that was recently published, and can we add to that understanding and really dive deeper into what are these sort of proportions of these immune cell types across these patients? The third sort of aim will be how can we identify from these sort of network co-expression modules, can we identify very specific pathways within modules that are specific to the patients with the sort of stem cell transplantation difference. So really diving deeper into a specific module that we're able to see whether that's enriched in a particular group and performing further enrichment analysis based on very specific regulatory pathways. So that will be sort of the three main focuses for today. The data set that we'll mainly be using and the experimental design background of the study is this specific data set within the Smart Bulk application. So if there is an interest in also doing your own analysis on this study, you can find it within Smart Bulk, which I'll show at the moment. So within the ecosystem, if we go directly to the Smart Bulk application, it'll open up the main interface when it comes to uploading your own internal bulk RNA-seq data. So for example, if you create a new project, that will sort of give you the information that you need in terms of what files you need, where you're uploading that data, and whether that data set is specifically a type of technology that you're using as well. So this is one way that you can access SmartBulk. The way that you would be able to see what public data sets we have currently curated within the SmartBulk repository database is through the public database sort of tab This will allow you to see what data sets we currently have that are accessible to you. And there's different ways that you can analyze this data. One where you can search for the individual study, or you can filter based on the ontology that is sort of mapped to these data sets to be able to create a custom atlas across different samples from these different data sets in the SmartBulk app. So this is quite useful, especially if you're focusing on adipose tissue and normally you don't have that many studies, particularly looking at this area. You can actually integrate those studies into sort of a custom data set and being able to analyze further samples than just sort of using one data set at a time. So that's something that you can also do to be able to really identify those studies in the database. Okay. In terms of the study design, these are the different groups that have been sort of included in the data set. So I guess the standard acute respiratory distress syndrome sort of characterized group, the ARD sort of group post stem cell transplantation. So those patients that don't generally have already an infection or came to the ICU with specifically this lung injury, but had it post the stem cell transplantation treatment. And we'll have other groups like sepsis, which is usually used as a control for looking at dysregulation immune response. So this will be the groups. This is specifically a study looking at ICU patients where they collected whole blood samples. And they also had different treatments that were also used within the study as well. So if that's something that you are interested in really identifying enriched pathway differences within these different treatment groups across these different samples, that's another analysis you can also perform. So the first step that I want to cover before we go into the data set and actually go through the first aim and the first research question that we're trying to answer, you have to keep in mind that when you're analyzing public data sets in the SmartBulk application, you have to make sure that any additional analysis that you're doing, especially if you're not using one data set, so if you're integrating multiple samples across different studies, you have to make sure that you perform your own quality control check and your own normalization steps on that data set. So that's something that's really important, especially if you're running weighted gene network co-expression analysis on sort of a public available data set. You have to make sure that these sort of samples are normalized and that there's a quality control check of the samples. So an example from this acute respiratory distress syndrome sample, when you go into the specific study that you want to analyze, for example, when I open up the data set in the SmartBulk application, I'm able to get details into the original metadata from that study. So that, for example, you can see how many samples you have, the anatomy information, condition information, and other metadata that comes with the study when it's curated. You're able to create composition or proportional specific visualizations as well to see sort of what cell counts do you have across these samples, and are you able to really perform a specific analysis to answer your question. And the really important step is really being able to go back and see if the original data set actually had these control steps in place. So did they have any QC parameters? Were any samples that had low quality transcripts sort of removed from the data set to be able to really have sort of a more concrete inferences from the results. So when we go into the study, you'll normally have the original PCA. So this currently is sort of the data set without any QC or any normalization sort of filtering. If you do want to perform filtering to be able to have a better sort of normalized count blocks plot to show that there's no distribution differences across the samples based on sort of their count matrices, you can actually go into the filter tab, and this will allow you to add different filters, different normalization sort of pipeline methods on the data that you can actually use to run further analysis. So one thing that I'll do is, since this data set did not particularly add any normalization methods and they didn't really apply any filtering, and we can see that by looking at just the count blocks plot across the different samples. What we want to do is select a very specific normalization method that is able to control and correct for these sort of matrices. So one of the methods that I'll use today is the TMM normalized method, which is a bit different from just applying log 10 normalization. So what it does, it normalizes generally across, or it transforms not only the raw read counts, but what it tries to address is the compositional biases and any differences in the library size, as well as effectively sort of accurately looking at differential gene expression analysis after this sort of filtering method. So that's the method that we'll mainly be using. Once we add the normalization method, there are other steps that you can also perform. So if you have multiple samples from different studies from the database, you can apply sort of batch correction on those samples as well. But in this case, I'm only using one data set, so I'm not integrating different studies from the SmartBulk sort of repository. So if I want to update sort of the plot, that will sort of run its own normalization. So I'm able to see the differences in the PCA once we apply that method, and I'm able to see if that actually sort of improved the count blocks matrices across the different samples. So we are able to see that it effectively was able to really sort of add to that raw count difference, and also that sort of sample variation as well. Okay, so that's the first step that I wanted to make sure that all the users that are using the application are able to really use this specific pipeline workflow step, even on your own data as well, if you haven't performed any of these QC steps in the study. Okay, so from here, once we have the normalization, we want to make sure that you subset the data. So that creates a new copy of the original raw matrices so that you have the normalized sort of sample data set that you're going to work with when you're applying all the different functionality analysis. Okay. So I'll go here and you'll see here that this is my original study. So the non sort of QC original data. And here I have these different sort of normalized data sets that I'll be using for the sort of downstream pipelines. Okay. So I'll go back and I'll select the last normalized data sets so that you're able to see it. So now that we have the data set sort of integrated, we can then move forward to the next step in the sort of agenda. So the first question that we want to answer is, are there any gene co-expression networks that are really unique to these patients that develop acute respiratory distress syndrome, so lung injury, post the sort of stem cell transplantation treatment, that were not initially captured by sort of simply applying differential gene analysis? So if we go into what the original data set did, so what this method will try to really do is really look at how can we construct a gene co-expression network to identify different modules that are correlated based on genes that relate to external traits. In terms of how this is applied, so normally the WGCNA uses the input from the count matrices. So you can actually use the normalized count matrices once you apply your normalization method. And this will help you to really identify the top 10,000 highly variable genes within the analysis, and specifically genes that reflect a very specific biological meaningful pattern that might be distinctive based on their co-expression sort of structures. So this will really be sort of the focus, and really looking if there's any hierarchical clustering that really identifies very specific gene modules across these different patients. Okay, so this is sort of a example of what sort of visualization and plots that the sort of method will be generating for you. So the main sort of focus is really to identify if there's any genes with similar expression sort of tendencies that are functionally related. So can we see that across very specific patient groups and even different treatment groups as well? Okay. And the other sort of, I guess, main point of the method is, can these modules group these specific genes with these similar expression patterns? Okay, so I'll go into the analysis platform so I can show you how you are able to perform that. So for the WGCNA, you'll see that this sort of functionality is currently in beta version. So that means that we're still developing the functionality further, and we really sort of depend on the feedback from researchers that are actually using this on their own data and that have any feedback in terms of other developments that we can do to make the output more clear, or the types of visualizations that you're able to export, and the type of data that you're able to generate as well from this specific methodology. So from here, the first step that you'll need to do is add an analysis name. Normally, as a default, it'll add the functionality that you're sort of performing. In SmartBulk, it'll add the sort of date when you're performing the analysis, and also the timestamp. So you're able to keep track if you're running multiple sort of comparisons. You're able to see which ones were run at what time and able to go back and then see which one you're actually going to use for your own analysis. The next step is selecting the data set that you're using. So whether you're using the raw matrices or the actual normalized matrices after normalization. So normally our team would really recommend, especially if you're running this sort of network analysis that tries to really construct the top highly variable genes, you want to make sure that the samples are normalized before doing that. Otherwise, you'll have sort of less sort of significant results, or you'll have sort of a different output than what you would normally have if the data is normalized. So that's something to keep in mind. Once you select the specific data set, you're able to run that WGCNA output, and that will sort of run in the backend. So it'll sort of let you know once it's sort of finalized and you're able to see those results. So I do have a previous result here that I want to show. And sort of the results that we're able to gather from sort of this WGCNA input. So some things that we can see just from looking at what type of modules we're able to identify. So the sort of the similarities across these different groups. So we're able to see that in this sort of module-based clustering, we're able to see some epigene similarity that shows some relationships to the type of modules in the different groups. And if we dive deeper into what are these epigene sort of co-regulation similarities across the different patient groups, we're able to see very distinct differences based on the actual modules that have this functional similarity. So this is something that you'll be able to generate across the entire data set. So you're able to see what are the modules that we're able to identify, and are these modules very specific to the actual treatment groups or the actual sort of disease models that we're analyzing in the study. So if I go back into here, what can we see? So we can see that there are some modules that are activated in ARDS that are transcriptionally sort of opposite to those in transplant recovery. And again, these are groups of genes that have similar expression patterns. So how can we see that in a deeper context? So when you're in the epigene sort of tab, which shows you sort of the distribution and sort of the clustering hierarchical heat map, you're able to also switch to the bar plot to see specifically what are the gene targets that are driving these different modules. So the background that is across the entire sort of hierarchical clustering, and also based on the very specific modules across the different patients. So what are these targets that are actually driving these functional sort of biological patterns? So if you planning to create a very specific module trait analysis that only focuses on, let's say, the disease model or the different treatment groups within the data set, you can go into creating a new module analysis, which can focus on a very specific metadata category. So in this case, I don't want to do it based on sort of sample variation, but what I want to do, I want to add the different conditions, and I want to also see if there's any differences on the treatments that are sort of conducted or that are treated within this patient group. So once we add this specific module trait analysis, that is sort of an additional step to the original module enrichment, we can then go deeper into actually looking at what are these specific differences across these modules. And let me just reload this so you're able to see it clearly. Okay, let me just go back here. Maybe it's just not loading currently. So if I have this specific copy of it as well. So what we can see when we add these module trait enrichment sort of results, we can see that there are clear differences in terms of the modules that are actually reflecting very specific characterization or different enrichment patterns within the ARDS group that had this sort of post stem cell transplantation. And we can see that it's very specific to a specific module. So we can see that specifically ME3 has a very specific enrichment within this group, as well as the acute distress syndrome category as well. So some of the, I guess, insights that we can gather is that the module, which is sort of specifically ME3, has a very specific immune context that is transplant associated, and has some likelihood of this sort of neutrophil reconstitution, or there might have cells that have this immune tolerance within these different patients. And this likely reflects how the inflammation is controlled across these different patient groups and sort of cell repopulation. So these are some insights that we can gather, especially when you're adding these module trait differences and how that relates to, for example, if we look at ME1, we can see that these modules are specific to the acute respiratory distress syndrome. So really looking at innate sort of ARDS inflammation that are dominant, that have this acute stress response, but that are not directly sort of correlated to the transplantation treatment, that might have some effect on these other patients. Okay. And we can also gather that if we compare, for example, ME2 or ME6, we can see that this is mainly a module that is related to these mildly anti-correlated sort of ARDS patients. So the sepsis control patients that do show some dysregulation, but don't really develop this lung injury model. So this might show another type of regulatory immune component that might have other cell types involved within these different patients as well. Okay. So the next step is really diving deeper or adding to this analysis that we're able to gather from the weighted sort of network co-expression analysis and really look at it within these sort of different modules that we were able to identify. Can we perform further enrichment analysis to really look at canonical immune or metabolic pathways that are disrupted within these patients post stem cell transplantation, and how these relates to sort of the standard sort of ARDS profile within these different modules that we were able to really define across the network analysis co-expression group. Okay, so if we go into our data set, the step that we would need to do, so when we have our WGCNA results, we can go into the sort of epigenes, so the specific module trait analysis, and we can also see this across single modules. So what are the genes that are actually driving these functional single modules? And we can select within these single modules a specific group that we want to run further enrichment analysis on those profiled marker signature genes. So one of the examples here, if we're interested in really going deeper into the ME3, which was specific to these patients that developed this acute respiratory distress lung injury, but particularly post stem cell transplantation, we can dive deeper into these sort of regulated genes, and we can perform further enrichment analysis. So this will allow you to really look at what are the further biological processes that we can define. Is there any cellular components that we're able to define within this specific module in these patients within this module, or even reactome pathways as well. So this is something that you can choose depending on what is the sort of geo pathway system that you want to really identify, and what is the module that you really want to sort of perform further enrichment on, or what are the co-regulated genes that you want to focus on. So an example, if we do sort of over-representation enrichment analysis, looking specifically at pathways in the reactome system, what we can see is that they're very specific pathways that are giving more insights into these differences across these different groups. So what do we see from just looking at these pathways? We can see that there's a higher transcriptional and translational activity, particularly looking at signals relating to proliferation and protein machinery that might sort of lead into this recovery post stem cell transplantation that might form more granulocytes. We can also see that there's a match in sort of the mitochondrial metabolic activation as well within this ME3 modulation, or this module from the network analysis. And we can also see that ME3, since we did select this specific module, captures sort of a different post-transcriptional regulation and cell death control. So these are the different sort of pathways that we're able to really gather. And you can also change the FDR score to really see these clear pathways a bit deeper, and sort of focusing on very specific significant pathways within this network module. Okay, so what does this mean in the larger scale? So we can see that ME3 as a module that has been identified to be specific to this ARDS post stem cell transplantation group. We're able to see that this specific module has these core transcriptional programs when it comes to the reactome pathway system that have sort of this immune dysregulation, and this might lead to differences in proportions in granulocytes that might likely be reconstituting different cells in the blood. It might mean that there's any metabolic priming or an antiviral response post-transplant. So these are some, I guess, key analysis that you can sort of gather. You can change the sort of visualization plot as well if you want to see it in a different scale, and sort of export those visualizations from the system as well. So normally the system allows you to add different settings, allows you to really interact into how you can customize a table further if you want to add any other information before exporting that data from the system. Okay. So I'll go back into the slides here. So the last question that we want to answer is how can we really define changes in immune cell populations, especially when you're working not on single cell RNA-seq resolution data sets, but you're working with bulk RNA-seq sort of sequencing. How are we able to really define immune cell populations from these samples by applying deconvolution methods into these public data sets? Okay. So some of the results, if I want to show you how you're able to perform that. So if you want to perform deconvolution on your data set, we do have that feature available within SmartBulk. And what you can do is that normally you have to select a reference database model to be able to apply that on your data set. So in this case, since the samples were derived from whole blood, I selected the blood sort of reference sort of name data set to be able to apply those specific cell type annotations on this study specifically. So that's something that you'll need to sort of define beforehand. We are working on being able to add your own reference data sets into SmartBulk, but that's a feature that will be coming later on as well. Okay. And one thing to keep in mind is if you're actually using a normalization method, so if you're using the normalized method that we performed, you have to make sure that you select that specific, I guess, version of the study to be able to perform deconvolution. Okay. So this methodology will take a few minutes to load. So I do have a previous result for this specific analysis that I want to show. So what can we see from these results? So we're able to see that there are different proportions of different cell types within these different patient samples. And what you can do is actually add different layers to that heat map or that clustering heat map. So you can see what are these sample, the patient sample variation. So is it specific to one patient or is it across different patients so that you're able to see if you have a, I guess, a level or a number of patients that is actually significant. You can actually look that through the heat map. You can add other clusters relating to the treatments that are actually used. So the post-transplant treatment or the bone marrow transplantation treatment difference. And you can see the actual sort of conditions as well. So what we're able to really define just by looking at sort of the deconvolution results is that we're able to see that these myeloid cells especially if we're focusing on neutrophils or monocytes, if we go deeper into the sort of genes that are expressed within these different samples, we can see that this really dominates across very specific conditions. So we're able to see sort of that these samples have this innate activation in these acute respiratory sort of lung injury model after transplant treatment. We're able to see that within these specific patients relating to this transplantation hematopoietic stem cell, we're able to see sort of a clear activation of these erythroid lineage cells. We're also able to see a higher proportion of myeloid leukocytes as well within these different patient groups that might be leading to these different inflammatory or immune remodeling differences. So overall, the deconvolution supports that there is a heterogeneous immune reconstitution state, particularly if we're just looking at ARDS patients post-transplant that are distinct to what we would normally see in a sort of acute respiratory distress syndrome sort of general model. And this aligns with the sort of interferon signaling that was sort of identified in the original data set from the author. Okay. So some of the summaries that we sort of discussed today when we applied these different three methods in SmartBulk. So the first sort of insight that we were able to see is that when we applied WGCNA methodology, so really looking at these sort of weighted gene co-expression network analysis, we were able to see and identify very specific co-expression modules that significantly were associated with profiles in patients that developed lung injury post stem cell transplantation. And this revealed a very specific network that was connected to genes that are likely sort of underlying these different transplant specific immune dysregulation sort of enrichment pathways. So this is something that we were able to gather when we performed very specific enrichment analysis on these modules that were specific to that patient group. We were able to see that there's specific pathways related to urocrotic translation, mitochondrial metabolism, and other pathways that are more related to RNA quality control as well. That might be leading to these different sort of expansion or granulocyte reconstitution within these sort of patients that have this innate immune activation. And the third sort of insight after performing deconvolution by using sort of the methodology in SmartBulk, we were able to reveal this sort of increased myeloid leukocyte dominance across very specific patient groups, particularly post-transplantation, and also a enriched erythroid and innate subset cell groups as well within these patient groups as well. So highlighting that there might be some lineage-specific immune differences that might alter the immune response within these different acute lung injury model groups. Okay. So I guess one thing that I want to highlight is that these clubs, again, are sort of conducted every week. So we're really happy to see any sort of researchers that have joined previously and that are consistently joining our sessions that cover different topics, different platforms in the ecosystem to really help you sort of gather what functions you can use to really develop your own sort of research workflow on your data. So if we have all the recordings that you can access on our YouTube page, and my colleague has sent sort of a poll that would be really useful to gather your insights on how this sort of exploration club session is useful. Were the sort of key topics or the key methods clear? And were you able to see how you're able to leverage these different methodologies for your own sort of data set analysis? So thank you again for joining this specific session. And again, one thing I want to highlight as well is that next week we will be taking a break from the Exploration Club because there are some holidays for the next week across our team. So I just want to say happy Reunification Day for our Vietnam team and happy International Labor Day as well. So thank you everyone for joining. And again, I look forward to seeing you on our next sort of Exploration Club sessions.